What is genetic engineering? Genetic engineering is a process of manually adding new DNA to an organism. The goal is to add one or more traits that are not already found in that organism. Examples of genetically engineered organisms currently on the market include plants with resistance to some insects, plants that can tolerate herbicides, and crops with modified oil content. Genetic engineering is a process designed to artificially control the genetic makeup of an organism. While genetic engineering has opened many doors filled with a plethora of possibilities in the world of science, from the fields of medicine, genetics, and biology to the realms of agriculture and produce, the effects of recombinant DNA and more gene manipulation are endless. Many types of genetic engineering exist from GMOs to recombinant DNA. One such implementation of recombinant DNA is the improvement of the cotton plant. Bollworms were becoming detrimental to the growth and the distribution of cotton crops. So, DNA from a bacteria known as Bt protein was combined with the DNA of the cotton plant and then injected back into the cotton plant. This allowed for the cotton plant to become poisonous to the bollworm. The form of modifications made to the cotton plant was transgenic. Take the vector DNA, which is good for the transporting enzyme, and coat with the restriction enzyme. Isolate the other portion of the DNA, insert the second portion of DNA in the vector. Next, insert the recombinant DNA in the organism cell. Now it is part of the cell's chromosomes. The ethics of science. Science never solves a problem without creating 10 more. George Bernard Shaw. Over time, as the field of engineering has grown, so have the number of ethical issues raised. The death of Jessie Gelsinger in 1999 is just one of the many failed attempts scientists have had in implementing still fledgling area of science. Her death was caused by multiple organ failures inflicted by a modified virus. Additionally, questions have been raised such as legal state of this field and success rate of this field. People have objected to the use of genetic engineering on several grounds, such as ecological and economical concerns raised by the fact that produce being grown along with or as GMOs possibly unsafe for human consumption and possibly other organisms to consume. Many benefits and concerns have been raised in the light of the recent popularity and necessity for the field of genetic engineering. One such benefit is the possibility of further beneficial medical advances for disease, viruses, and other medical conditions that have been problematic in the recent years. Success has already come from this field as recombinant DNA vaccines have been very successful. HPV vaccines and hepatitis B vaccines are just two examples of the success. Furthermore, the field of STEM research has been affected. An experiment finding if injecting stem cells in mice might reserve plaque buildup in arteries found interesting data. Female mice were given male stem cells, and male mice were given female stem cells. So, the Y chromosomes could be tracked. The results found that the male mice with female stem cells got better, while the female mice with men's stem cells got worse. This could mean that the female stem cells might reserve plaque buildup in other organisms as well. This was only possible, though, through the use of genetic engineering. Some of the concerns that have been raised involve the health of people as they consume genetically modified foods produced on farms. This could lead to health issues as well as an increase in the price of organic produce. Many people feel the use of genetic engineering in food and farming is wrong, and that it goes against nature or their spiritual beliefs. Now, others think that is wrong because it allows big companies to gain more control of the food chain. The fact is that genetic engineering allows scientists to take one gene from one species and insert it into a completely different species which could never naturally breed. The current application of genetic engineering is in agriculture. So wheat, corn, chicken, and beef are like the subject to kind of this tinkering. Because genetics are not completely understood, the long-term effect of changing the DNA of these foods cannot be known. People are concerned that these effects may be harmful for humans. Thus, thus endangering our food supply. In addition, there is a fear that such modified crops and species may get into the wild. Human history has had examples of plants and animals moving from one environment to another, causing immense damage. How much damage would a genetically modified plant or animal being introduced into the wild can cause? While genetically modified foods in America would have little impact on people in China, it is not hard to imagine the impact on both North America and South America, not to mention all of the countries America exports food to. A key part of any benefits discussion involves alternatives. Are there any better ways to do and get what is desired? Like benefits, discussions of alternatives can be complicated and elusive, and much depends on the goals envisioned. When the goals include reducing dependence on pesticides and herbicides, there are clearly alternatives to many biotechnological products. Many of these alternatives are not other products, but instead the systems and methods of sustainable agriculture. A good example is a crop rotation, which keeps pests under control elegantly by depriving them of continuous food supply they will need to build up large populations. Crop rotation has many advantages. It controls a broad variety of pests rather than just one or two. It does not select for resistance genes, as do chemical toxins or genetically engineered crops, and it does not result in ongoing pollution of air or water. As a pest control strategy, crop rotation is far preferable to both chemical insecticides and genetically engineered crops. Unfortunately, because it involves process and not products, there is no industrial consistency to develop and support crop rotation as there is for products of biotechnology.
What is the Human Genome Project? The Human Genome Project, or HGP, is an experiment with the task to break down the chemical base of DNA. This project is funded mostly by the U.S. Department of Energy and National Institute of Health. Genome. A genome is all the information coded in DNA, variation, mutation, genes, etc. The point of the Human Genome Project is to discover these variations, mutations, and traits in humans just by observing their DNA. The process has made it possible to research and detect variations in human genome. Mainly disorders in people are caused by several complex variations in human genome, so it's still not possible for scientists to detect exact disorders in humans. Bioinformatics. Bioinformatics is the mapping of the human genome using computer science along with mathematics in order to detect different sequences in DNA. This is what the Human Genome Project uses to decode variations in DNA and find the mutations and disorders among humans. Pros and cons of the Human Genome Project. Pros are that people can detect disorders or illnesses by looking at their DNA. This would lead to stopping or catching disorders and illnesses in its early stages, and much more information on these disorders and illnesses and how they can be detected in DNA. Cons of the Human Genome Project is that it destroys the privacy of one's DNA. Once your DNA is tested, it is permanently in records. To what extent do people have access to an individual's DNA? Is it just hospitals and insurance companies? Or can your jobs, friends, family, or whoever access your DNA? Would people be able to choose who has access to their DNA? Also, this raises the question of will employers use discrimination when hiring? Employers will obviously want to hire people with superior DNA, and certain disorders and genomes that cause illness will cost insurance companies more money,